This lecture is being brought to you in part by the generous gifts of these sponsors.
Thank you very much, Ken. I am delighted to be here. I have had an absolutely amazing day. And I wish I could stay longer, but I hope at the very least I'll be invited to come back. Just a funny story before I begin about cell phones in lecture halls. I was once giving a lecture and didn't pay any attention to my phone, and I was about 10 minutes into the lecture, and a phone starts to ring. And the organizer and the phone kept ringing and ringing and ringing, and the organizer, timeout, whose phone is that ringing? And I just said, it's mine. So I had to come down off the stage and turn off my phone, but my phone and my purse are somewhere else, so if a phone rings tonight, I know it's not mine. So what I want to do in the talk this evening is give you an introduction to the human microbiome. And before I get into the talk, let me just see by a show of hands, how many of you have heard of or read something about the human microbiome? Feel like you have some? Yeah. I'm not surprised, because in the 40 years that I've been doing research, this has been, for me, the most interesting and exciting research that I've been doing. And it's also the research that I think has most captured the imagination of the public. And I think for good reason. So I hope, in part by the end of the talk tonight, I can convey to you some of the excitement that I have for this field of study. So let's begin, and I'm going to pose a question to you. I would like you to think of a thriving ecosystem. And so I'm going to guess that for many of you, when you think of an ecosystem, you think of a rainforest. For others, you may think of a coral reef, although I think if we look at what's in the news, we might say that these ecosystems aren't particularly thriving anymore. This is an interesting example of a thriving ecosystem. This is Mr. Owens. He lives in the UK. He planted this terrarium about 30 years ago, sealed it up, and it's been doing just fine. So that ecosystem has everything that it needs. But what I hope I can convince you of tonight is that we, as humans, along with our microbial partners, represent a thriving ecosystem that is just as interesting and just as important and just as worthy of study of any of these other examples. So these are some of the topics that I'm going to cover tonight: how we study the microbiome, what we know, the implications for the microbiome in chronic disease. I'm going to talk about the gut-brain microbiome axis, because I think this represents the forefront of the field, and finish up with a few thoughts about how we might deliberately manipulate the microbiome to enhance health and maybe treat disease. Certainly, if there are questions, we can go beyond any of these topics in the Q&A period, but this is how I plan to structure the talk. And just two other items here in terms of terminology, and sometimes these are used interchangeably, these two terms, but they really shouldn't be. You'll oftentimes hear someone refer to the microbiota. What this represents is the collection of all of the organisms that share our body space. Microbiota is a collection of organisms, whereas the microbiome is the collection of all of these organisms and their collective genetic material. So just a minor point, but if you hear me use these two terms, hopefully I'm using them correctly, and I'm using them for a reason. I'm either talking about the organisms or about the organisms and their genomes. And as is so often the case in science, even though a lot of intense focus on the human microbiome began about 10 or 12 years ago, we weren't the first to do this. In fact, the first description of the microbial life that's found in association with humans was made by Antonin von Leeuwenhoek. One of the very first samples he looked at after he invented his compound light microscope — he was the inventor of the microscope — was some of the material that he scraped off the surface of one of his teeth. And he suspended that in solution on a microscope slide and looked, and what he saw just amazed him, so much so that he wrote about this in a letter to his colleagues at the Royal Society of London. This was the first time, and really think about how profound this was. This was the first time any microorganisms were seen, because before the development of the microscope, they were invisible. No one was calling them bacteria at the time. Von Leeuwenhoek called them animalcules, but he noted that they were present in such enormous numbers that the water seemed to be alive. And this is interesting, and we've really come full circle 350 or more years later, because the oral cavity has been one of the sites, one of the environments in the human body that has been the target of intense study as part of the Human Microbiome Project. So I would say that for the better part of that 350, 360 years, the human microbiome, the organisms and their genetic material, were really still very much beyond the reach of our understanding. Other than being able to see these microbes, there wasn't really much else that we could learn about them. And for a very long time, it was thought that these microbes, this collection of microbes, which had been called flora, or the normal flora, for a very long period of time, it's thought that they were inert. We were just carriers, that these were relatively stable over the course of a lifetime, that they weren't at all relevant to disease. They were just, you know, sort of like dust particles that we were carrying with us. And I think to a considerable extent, even within the field of clinical microbiology, pathogens, disease-causing microbes, were really thought to be the ones that were most relevant to human health. And I would argue that even still, in medical schools around the world, the first introduction that students have to microbiology is in the context of disease-causing organisms, not the organisms that we share our environment with from the day we're born until the day we die. And certainly at Maryland, my colleagues and I are trying very hard to get the curriculum changed, because I think that in the not too distant future, we're going to see that measurements of the human microbiome in one way or another will start to become part of the standard of care, much like you think if you go to the doctor now for a physical, he or she will draw blood and measure all of your blood chemistries. So it was about 2005, 2006 in this country that an effort out of the National Institutes of Health in Bethesda was launched to put together a collaborative project involving investigators across the U.S. to study the human microbiome. Why then? Well, I think in large part it was because the tools of genomics that in large part had been developed to complete the sequencing of the first human genome were equally amenable to the study of all of the microbes that we share our space with. And quite frankly, all of these big centers and smaller centers like my own that had been set up to do some of this work were now sort of looking for a next big project. So after a number of planning meetings that I participated in, a roadmap for the Human Microbiome Project emerged. And one of the primary goals of the first 5 years of this project was to use the available tools that we had to try and understand what a normal microbiome looked like. And that may sound easy in principle, but as I hope I can explain to you, it's been a little bit harder in practice. Normal might not be the correct descriptor. Maybe it should be healthy. But then we got into discussions, well, what does healthy mean? Is health just the absence of disease, or is health something more? And so not only were there scientific challenges, but we're still struggling with some of these philosophical challenges. But what the Microbiome Project did was enroll about 300 healthy volunteers between the ages of 18 and 45, and I have very sad news for all of you in the audience over 45. With a lot of scientific studies, once you pass 45 years of age, you're considered elderly. I'm in that same category. It's hard to take. And these volunteers then had to agree to donate samples from multiple body sites over multiple periods of time, and the scientists involved in the project set about characterizing what these collections of microbes looked like from each of these individuals. I was also involved in a couple of projects. There were a total of 15 projects to investigate whether or not we could identify some sort of change in the microbiome in various human environments, either the oral cavity or the skin or the GI tract or the vagina, whether we could identify changes associated with various diseases. These were called demonstration projects. For obvious reasons, they were meant to demonstrate or not that there might be a link. This is the only slide like this that I'm going to show you, just because so much of what we know to date is based on this. And this is using a particular gene that you find in all bacterial species that's part of a key component of the machinery in the bacterial cell that makes proteins. And what we can do with this is specifically target regions of this gene called the 16S ribosomal RNA gene. And we can generate DNA sequence data from portions of this molecule, and we get a readout from every single cell in the community that we're studying. And what I like to do is think of this kind of information as being absolutely analogous to a barcode. So the way we analyze this data is we generate all of these barcodes. It's very straightforward. We can process hundreds or thousands of samples at a time. And then we count. How many of this barcode do we see? How many of another barcode? And the number of barcodes that you can capture is really infinite. So what we're doing and what the field did for the better part of the first 5 years was really use this approach to take a molecular census, to count how many of this type of organism, how many of that type of organism. And much of what you read in the lay press to date is based on these kinds of measurements. And I would just caution you, while the stories that you read can be very exciting and suggest very definitive links between changes in the microbiome and disease, what this information doesn't capture is anything about what these organisms are doing. So again, if you think about it, analogous to a census, here you've captured the name. Think of, if you've participated in a census, and I go on Ancestry.com and look at, what my grandparents and great-grandparents did. Other information will tell you what their occupation was, what their age was. It gives you a richer view of the community, whereas this is just a name, but it's a pretty good start. So, as I say, one of the most important first objectives was trying to define a healthy human microbiome. My environment of interest is the gut, and everything that I'm going to talk about tonight has to do with the gut, but the ideas, the approaches are equally relevant to any of the other human environments. And so part of the reason why defining a healthy gut microbiome was so important is that if you know what's healthy, then you can better identify a diseased microbiome if you see one. And if you know what's healthy, then perhaps it gives you some ideas about how you might deliberately intervene to move a diseased microbiome back to a healthful state. Does that make sense? Great. Okay. And so what we found, somewhat to our surprise, was that when you looked across the human body, as a result of all of these analyses, we immediately see that we're not a singular environment. We're not a single ecosystem, if you will. Depending upon what portion of the body was sampled, and these pie charts represent relative abundances of different types of bacteria, their identities don't matter so much, but what's important is that you see no two pie charts look the same. And additional studies have been done looking at the skin, for instance, and revealing that if you're right or left-handed and you sample the right or the left side of your body, you can actually find differences in the skin. So the variability is phenomenal, but that's really in keeping with what you would expect in a complex ecosystem. Not every niche is exactly the same. They may be related, but they're not exactly the same. So what's there, and what do all these microbes do? Some of these numbers just blow me away. It's estimated that there may be 100 trillion bacterial cells that we carry with us as adults. You make some calculations and some assumptions, and we quickly come to realize that those 100 trillion cells, which are made up of thousands of different species of bacteria and fungi, carry 10 times the number of genes that we think are encoded in the human genome. That's a lot. So just on the fact of sheer numbers alone, I think we have to pay attention to our microbiome. But what we've also come to understand is, as I said earlier, these aren't just passive hitchhikers, if you will. They're essential to human health, and they're involved in some really important biological processes. They help us extract energy from certain kinds of food, and I'll say a fair amount about this in just a few minutes. They produce essential vitamins that we can't make on our own. They regulate our immune system starting very early in life, and it's thought that the increase in certain diseases like asthma and food allergies and eczema, diseases that are thought to have perhaps some sort of autoimmune component, may be the result of inappropriate crosstalk between the microbiome and our immune system early on in life. They've been shown to regulate glucose levels and also be involved in other aspects of our metabolism. And I think this is a really important role here. They protect us against disease-causing microbes. I've been doing some interesting studies with a collaborator at Maryland looking at, both in humans and in some non-human primate models, looking at infection with Shigella and Salmonella that are two enteric pathogens. These are pathogens that cause food poisoning. They're particularly of public health concern in the developing world. And we can find subjects who can take a highly virulent, highly infectious dose of either of these two pathogens and not get sick, not develop a single clinical symptom. And you probably know people, if you travel, you probably know people. There are some people that get sick the moment they touch down on foreign soil, and there are other people that can go through an entire trip and eat exotic food and never get sick. Well, we've actually, again, found some interesting correlations. It doesn't prove cause and effect, that some of these, that the people who seem to be protected have a different microbiome in their gut than the people who get sick. Well, that's pretty cool. Maybe we can exploit that for those of us who travel. And I think one of the real surprises that came out of the first phase of the Human Microbiome Project was the finding that there is tremendous inter-individual variation in the microbes that we share our space with. So much so that even in a given individual, from one day to another, these communities may look to change. But if you look across individuals, and certainly if you look across populations around the world, you can see some pretty major differences. And we're just beginning to explore this, and we'll talk about this now in a bit more detail in the next phase of the talk. We know some of the factors that we think influence these changes in the microbiome, and the two that I'm going to talk about are diet and antibiotics. Those have tremendous effects, but there are other effects. We know that stress, certainly chronic stress, can have an effect. Infection with various pathogens. And I have a colleague at Maryland who's trying to convince me to get involved in a study because he believes exposure to air pollution may somehow alter the structure or function of the communities in our guts. So I certainly now have a much more holistic view of health and disease, and I think that the data are absolutely pointing us in that direction. A lot of emphasis has been placed on trying to understand the changes in the microbiome over a lifespan. So there have been studies looking at what's in utero, looking at newborns, babies, all the way through to the elderly. And we know now, going back, thinking about these as ecosystems, that in the very young, in infants, these communities are fairly low diversity. They're changing all the time. And at about 3 years of age, they stabilize and take on the structure and function of what we see throughout most of adult life. The mode of delivery, whether it's vaginal delivery or C-section, has an effect because we inherit our microbiota from our mother. So we're either getting our inoculum through the birth canal or from the skin. And these infants have been followed for the first couple of years of life, and these differences that you see at birth persist. And that raises an interesting question. Do they ever converge? And if not, is there a consequence of that? And breastfeeding versus formula feeding have also been shown to have really profound effects. And in fact, some of the key components in breast milk that formula companies have been trying for a very long time to reproduce are the oligosaccharides that you find in breast milk. Infants don't have the mechanisms to digest these oligosaccharides, and it appears that they're there to feed the new microbes in the infant's GI tract. So talk about a coevolution of a whole system. Once we move from adulthood into old age, and here it's shown as 65 to 80, we've seen that these communities tend to lose diversity again. And think about ecosystems. You think about a healthy ecosystem is one that has a lot of biodiversity. So if you start to lose diversity, maybe there's a potential consequence. And I can tell you, the worst possible situation to be in is an elderly person in a nursing home. The diversity in those cases, in a couple of pilot studies that have been done, is almost non-existent. And the reasons for that are not entirely clear. So we talked about age, but there are a lot of other factors, and we'll move right on because I've mentioned these already. Diet and antibiotics are the two that I'm going to focus on tonight. So let's take the next few minutes and think about diet. This is a schematic diagram of our GI tract, starting at the stomach. And what you see here in red, green, gray, and blue are the different types of microbes that you find across the length of the GI tract. And we also have a couple of different kinds of nutrients shown here. Simple nutrients that we as humans can digest without any help from our microbial partners. And what's labeled as undigestible nutrients, this string here, this string of beads, which essentially represents fiber. We can't digest any of the fiber that we eat on our own. It's through the actions of the microbes in our GI tract. So by the time food gets through the small intestine, which is shown here, most of these nutrients that we can digest, proteins and fats and simple sugars, are gone. And what ends up being delivered to the large intestine are all these undigestible nutrients, where a completely different collection of organisms goes to work and starts to digest this fiber. This is a critically important process for health. And I hope that the next slide gives you some idea why. I actually have this slide right next to my desk because I have to refer to it all the time to try and make sense of my data. But what it's meant to show, don't worry too much about this, but what it's meant to show is that there are different groups of organisms that we know have the capability to metabolize different types of dietary constituents. So here we have dietary fiber and mucus, dietary protein, dietary and host phospholipids or fats. And these are the input. The bacteria do their thing, work magic, and various metabolites are then produced. And I put this up here just to make a couple of important points. These butyrate-producing bacteria in the large intestine are critically important for health. And you can see that what we have to feed them is dietary fiber. And if we do, they produce these metabolites called short-chain fatty acids. Butyrate, I would say, belongs on the list of wonder drugs or wonder metabolites. It helps to nourish the cells that line the GI tract, and I think perhaps more importantly, it has been shown to have potent anti-inflammatory properties. The only way you will get butyrate produced in your GI tract is if you feed this group of microbes, and they need fiber to be happy. So I am certainly guilty as anybody, where days or weeks used to go by. I've changed my eating habits, but days or weeks would go by when I didn't consume particularly much fiber. So you might ask, well, what happens if that's the case? These microbes are still there. They need to be fed. And if we're not feeding them what they need, they start to feed on us. And on the top of the cells lining the GI tract is a thick layer of mucus. That's typical of epithelial cell layers. And if these guys don't get what they want, they're pretty darn happy eating that mucus layer. And you can see in patients with a lot of GI diseases that this mucus layer in some places is completely gone. And that leaves a vulnerability. That exposes these epithelial cells lining the GI tract to everything else that's going on out here. We'll come back to that message. That's an important message that I'm going to be delivering. This is another interesting part of the story. Here are some of the bacteria that digest fats. They can either be dietary fats or phospholipids that come from breakdown of cells in the human body. But if you think about a key source of dietary fat, it can also often be animal fat. You know, who out there doesn't occasionally like to eat the big steak? I was with you. That was me. Bacon. Thank you. Bacon. Well, what these microbes then have the capability of doing is taking that input and metabolizing the fat to produce compounds like trimethylamine and ethanolamine. And these compounds have been shown unequivocally to promote atherosclerosis. So I think we know that eating a lot of red meat is associated with atherosclerosis and cardiovascular disease. What you maybe didn't know is that that doesn't happen directly. It happens through the action of some of the microbes in our gut. Are you starting to reconsider what you eat? Maybe I haven't convinced you yet. This was a really interesting study published a few years ago by Peter Turnbaugh's lab at Harvard. I just take a figure from this paper, but what he did was take a group of volunteers and feed them, I think it was for 4 weeks, on a plant-based or vegetarian diet, and then switch them to an animal-based diet. This is looking, I think, fiber, overall fiber intake, fat. I can't read that here. But the point is that as the constituents of the diet changed relatively quickly, so too did the relative abundances of these types of organisms that I was just talking about on the previous slide. So we know from these kinds of studies that if you change what you eat, you can either feed or starve the good or the less good bacteria in the GI tract. Bad news here is that a week or two of good eating habits doesn't last you forever. You need to keep going with it. And this again, I won't spend any time on this. This is just the same idea that what we eat, the different components of our diet, feed different groups of organisms in the GI tract, and we can either make use of that to increase the types of metabolites that promote health and reduce inflammation, or conversely promote things like atherosclerosis, deposition of fat, and in some cases cancer. So I get asked this all the time, and I'm not a nutritionist and I'm not going to say any more of this, but what are you supposed to eat? Well, probably a good place to start would be to think about a Mediterranean diet versus what I think is a typical Western diet. I flew in yesterday in the airport, and I certainly saw an awful lot of people between flights eating pizza and ice cream and greasy burgers and fries. And if these are the main constituents of your diet, and fruits and vegetables and whole grains are down here and or what you eat occasionally, probably not a good thing. Whereas this food pyramid really needs to be flipped. And I'll give you a recommendation at the end of the talk if you're interested in a book that gives you more information about this. But when you talk about diet, I think an obvious question is, well, then how should I be eating? What should I be eating? And I'd say that this is a good place to start. So now, again, getting back to this concept of ecosystems, we know that ecosystems in general are very dynamic, and a stable ecosystem has two properties. The first is resistance, and that is shown here by the blue line, that even in the face of different disturbances, whatever they might be, the ecosystem function is maintained. You might reach a point where one more disturbance causes the ecosystem to crash and burn, but in general, a resistant ecosystem is a healthy ecosystem and can maintain its function. But even if that doesn't happen, there is another ecosystem property that's important, and that's the idea of resilience. And that is shown here by the red dotted line. That's the ability of an ecosystem to absorb a disturbance and recover, retain its basic structure and function. So you could think about it like this: function, something happens, function changes, but then it comes back and maybe happens again and again. And this, I think, becomes very, very important when we think about ways that we may be unintentionally altering our microbiota. This is, again, just another diagram. Here's a healthy system. Everything's in balance. You have an intact epithelial cell layer in your gut. Here are the microbes in the microbiota. You might have pathogens coming through, but the system is such that they don't really cause much damage. Contrast that with a disrupted community, where you have loss of integrity here in the epithelial cell layer. These pathogens come in and can overgrow and cause problems. What's in the lumen here gets into the circulation and can cause systemic infection. And probably one of the easiest ways to trigger this kind of disruption. And this kind of disruption very often then leads to inflammation. And one of the easiest ways to trigger that is through continued exposure to antibiotics. And what's interesting, again, I'll sort of a show of hands. How many of you out there can remember how many times in your life you've taken antibiotics? Wow, that's more than I would think. Because for a lot of us, we don't even remember. Certainly as kids, I don't know if I was given antibiotics much, if at all, as a kid. I suppose I could ask my mother, but this idea that antibiotics disrupt the microbiota and lead to changes that can promote inflammation, I think is supported. Again, it's not cause and effect, but it's supported by a little bit of research that I did. I was reading this paper unrelated to the microbiome that had to do with chronic inflammation and a number of diseases. And I know this isn't easy to read, but these are sort of all the chronic diseases that we face. And as I was reading that, I thought, gosh, I think there's been a fair amount of microbiome work done on a lot of these diseases. So I did literature searches, taking each of the diseases one at a time, and went into Medline, which searches the scientific literature, and typed in each one of these diseases and microbiome. And in every case where you see an asterisk, I found at least one, and sometimes tens of papers, reporting links between changes in the microbiome and these various diseases. And what all of these diseases have in common, and what was the point of this particular article, was that there's a component of inflammation associated with each of these chronic diseases. So we need to be careful here, because these are just associations, and association does not mean causation, but it leads one to wonder, is there a common theme emerging here? All these diseases that have inflammation as a component, and the loss of a healthy microbiome that can be associated with inflammation, it makes you start to think, are we inadvertently altering our microbiome in such a way as to promote these inflammatory diseases? And I think that we have to pay particular attention to the potential role of antibiotics in driving the evolution of our gut microbiome in that direction in a way that we don't particularly want. There have been some fascinating studies done looking at numbers of different types of antibiotics in volunteers, and we see after antibiotic administration two things happen. One is that we see a real enrichment for antibiotic-resistant genes. That's not good. But we also see, and I think this shows on the next slide here, that, and these represent the results of multiple studies looking at antibiotics. These bars show you percent decrease in biodiversity in these communities, in these gut communities after exposure to antibiotics. And in all cases, there's a decrease in diversity, in some cases a significant decrease in biodiversity once we take these broad spectrum antibiotics. They were designed to go after disease-causing bacteria, and when they're medically indicated, we absolutely need to take them. But the problem is that there can be collateral damage. And this is an example here. And if you look at what happens in these patients following discontinuation of antibiotics, in some individuals, the communities restore to the state that they were in prior to antibiotics. But in all of these studies, there was a subset of individuals where the structure and presumably the function of the microbiota seems to be irreversibly changed. So if you think about that happening over and over and over again over the course of a lifetime, we may be inadvertently driving the evolution of our microbiome to a less diverse state. And this is work from my friend and colleague, Marty Blaser, who's at NYU. And he has proposed the idea that if we look at the blue line here, this is an example of what might be happening in the United States. It goes back, almost 100 years. So these are not real data. This was an op-ed piece, if you will. So it may have been that 50, 80, 100 years ago, we had a microbiome that was considerably more diverse. But through continued exposure to antibiotics, both medically indicated and in our food supply, the increasing ingestion of more processed foods, we're starting to see a loss in the diversity of our microbiota. And remember, I told you that we inherit our microbiota from our mothers. So there's no real way to reset the diversity of the microbiota once it's been lost. And this isn't just total speculation, because if you look at some data that Marty has also collected from populations that live in the Amazon rainforest, for example, that these are populations that are essentially untouched. And you compare the diversity of the microbes in their guts to what you would find if you sampled everybody in this room, and it's dramatically more diverse. So it doesn't prove anything, but it's consistent with this idea that modern life may be moving things in a direction that we don't want. And so one of the really critical questions is, and this is almost in some ways analogous to the climate change discussion, is there a way to stop this decline, and can we figure out ways to start to reverse this trend? And why do we care? Does this really matter? Well, again, Marty's done a number of studies in mice looking at the effects of antibiotics, and he has shown that when you change the structure of the microbiota with antibiotics, you also change the body composition of these animals, and they start to accumulate fat. This promotes what's in humans is called the metabolic syndrome. It's a pre-diabetes state, and genes involved in immunity are also downregulated. And I will be the first to tell you that mice are not humans. But if you think about the increase in obesity and diabetes, certainly it's being driven by dietary changes over the last several decades. But this could be contributing as well. And again, what also is consistent with this idea is the fact that in the livestock industry, antibiotics are used at subtherapeutic concentrations to promote growth. So again, these are intriguing ideas, and they're linked together, but we still have a lot of work to do to prove all of this. But there have been a couple of studies published, and the results are here. One was looking at a large cohort of children in Finland who received early life antibiotics exposure before 6 months, and then another study, even larger study in the United States, looking at 64,000 children, showing that in children that were exposed to antibiotics before 6 months of age, if they were then looked at at 2 or 3 years of age, those kids had a higher BMI than kids who did not receive antibiotics. It's pretty causes you, at least causes me to stop and think. So it may be that this is where we have ended up, not in any deliberate way, and we didn't have the tools to study the impact of things like diet and antibiotics on the microbiota until fairly recently. And so if you're interested in this in any more detail, I would highly recommend this book by Marty Blaser, Missing Microbes. This was really, it was written for an interested general audience. It's not a science book, and he puts forth some really intriguing ideas. So let me get on to one of the last topics, and I'm almost out of time here. I want to make sure there's time for questions. And I just want to make a couple of comments about what I think represents the new frontier in the microbiome field, and that is looking at the connection between the gut and its microbiome and the brain. And you might say, how could there possibly be links between the gut and the brain? Well, we know that there are lots of different links. The nervous system innervates the gut. The vagus nerve is sort of a superhighway between the brain and the gut. Microbes in the GI tract make compounds like dopamine and serotonin that are key targets for various disorders when they're produced by neurons in the brain. And the immune system also may play a role. So there may be some real links here between the gut and the brain. And it's really sort of turning the whole idea of understanding mood and behavior on its head, I would say, and getting us to focus on things outside of just the nervous system. Again, this is just a slide to try and reinforce the idea that there are multiple types of connections between the gut microbiota and the brain. And when things in the GI tract get out of balance, the impact may not just be locally in the gut, but there may also be an impact in the brain. So this slide here is really meant to summarize some of the studies that have been done to date. Many of these have been done in mice, and again with the caveat that mice are not humans, but mice are a whole lot easier to work with than humans, particularly for some of these kinds of studies. But you can think that, again, if we've talked a lot about a healthy microbiome, everything is in balance, everything is operating as it should be. But what we know is that if you, this equilibrium is perturbed, whether by stress or disease, you end up with increased levels of inflammatory mediators in the circulation, you have an altered composition of microbes in the gut. And what we've seen in a number of studies in mice is that when that happens, you also see very significant changes in mood and behavior. You end up with mice that are more anxious, mice that don't learn as well. And so, again, if it's true in mice, might it not also be true in humans? And I think one of the really interesting findings from studies on mice is that there seems to be a critical window when this disturbance is most impactful. I'm not going to get into details about these studies with germ-free mice, but the critical period in mice seems to be during adolescence. And if you think about it, there are certainly mental illnesses like bipolar disease, like schizophrenia, that don't manifest often until young adulthood. So it's not, it doesn't prove this in any way, but it's consistent with the idea that there may be some disturbance during adolescence that pushes things in that direction. These are going to be really tough studies to try and put together and get meaningful data, because you have the complexity of the microbiome, you have the complexity of the nervous system, and then you're really going to try and put these together. But it's where my research is now starting to go, and you know, I hope we and others can start to make some real progress there in understanding all of this. Final topic, final question. This will just take a couple of minutes. All right, we think we know, we may not entirely be able to define a healthy microbiome, but maybe we are a little bit better at identifying an unhealthy microbiome if we see one. So that raises the question, can we intentionally manipulate our gut microbiome to move a disease state back to one associated with health? I would say at this point there are probably 4 main approaches that we have at our disposal currently to think about doing this. Antibiotics, and we've just talked about why antibiotics are bad, but certainly antibiotics are one way to affect the microbiome. I put this up here more forward-thinking because it's not out of the question to think about developing species-specific antibiotics that might target one type of organism that you want to modulate. But for now, we don't have any of those currently, but that's a topic of discussion. Is it feasible? Can we do it? And I think it is feasible. I'm not sure if it will be done. Probiotics, prebiotics, and fecal transplantation. Let me ask, how many of you take probiotics? And I will put my hand up as well. That doesn't necessarily mean it's a good thing. But probiotics, I would say that, let me go to the next slide. Probiotics, there's a very specific definition of probiotics, and this was a definition that came out of a meeting at the World Health Organization back in 2001. And the working definition of probiotics is live microorganisms which, when administered in adequate amounts, confer a health benefit on the host. Seems pretty obvious what we're talking about here, but scientifically, to try and quantitate some of these things, it's a little bit difficult. What's an adequate amount and what's a health benefit? But there are lots of bacteria, and in one case, a yeast that you can find either in the dairy aisle at the supermarket or in capsule form. These are all organisms that originally were derived from microbes in the human gut, and they were originally used to ferment various foods as a way to preserve them. Probiotics weren't developed to improve health, but there was this association that was noticed that particularly in Eastern Europe, there were populations of people that ate a lot of fermented dairy foods, for instance, that seemed to have a good amount of longevity. And that's where this whole potential link between probiotics and health came from. This is an area that my lab is actively investigating. A lot of studies have been done, but I would not say that they've been done with the rigorous tools and analyses that we're now applying to the study of the microbiome, and a lot of that work is being repeated, so stay tuned. One thing that we have found, and I don't think it's all that surprising, is that if you look at a singular probiotic, it's unlikely that it will have the same beneficial effect in all individuals. And that shouldn't surprise us. We know that's true with prescription drugs, so why should it be any different with probiotics? But I think that this is a field that's ripe for progress now that we have these tools in hand. And this is the last idea, the idea of fecal transplants. Again, how many of you have heard of fecal transplants? Wow, this is a really good audience. Hopefully, I mean, I could ask this question, but I won't. You can go online and find do-it-yourself protocols for fecal transplants. I'm not going to ask how many of you have done that, but you can actually do that. We teach this to the medical students at Maryland, and there's a certain yuck factor about it. And at the end of my lecture, I always ask the class, okay, so if you needed a fecal transplant, who would you want to be the donor? And, and the most interesting answer I ever got was one guy said his dog. I didn't explore that further. But there is one particular indication where fecal transplants, and for those of you who aren't familiar with them, it's really just as it sounds. It's taking feces from a healthy donor and delivering them in either direction. They both seem to be equally effective to someone who is sick. And it's been shown to be highly effective and is probably very soon going to become the standard of care to treat antibiotic-associated diarrhea as a result of overgrowth of Clostridium difficile. This is a terrible disease. It will at times develop in patients that have been on chronic IV therapy with antibiotics, sometimes a whole cocktail of antibiotics. Well, it wipes out all the beneficial microbes in the gut and gives C. difficile, which is a normal resident of the GI tract, an opportunity to overgrow. And C. diff can make these toxins that will lead to terrible diarrhea. It's not just a disease that's inconvenient. People have died. People die from C. diff diarrhea. A couple of clinical trials were underway comparing fecal transplant from a healthy donor into someone suffering with C. diff diarrhea to the standard of care, which has been high doses of vancomycin. And these clinical trials were stopped because the difference in the cure rate between fecal transplants and yet another antibiotic was so great that it was deemed to be unethical not to offer this to all of the patients who were enrolled. And it's just exactly as it sounds. And again, it makes sense from an ecosystem perspective. You take a terribly disturbed ecosystem and reseed it with everything that should be there. And these bacteria have the ability to take hold and colonize. And it's amazing, within 24 or 48 hours in these people, diarrhea resolves. But there is a bit of a yuck factor. The FDA for a while was just saying this is a treatment of last resort, we're not going to get involved. But now they are starting to think that they really need to consider this, and is this something you think of as you would, say, in any other organ transplant? But a number of companies have been established that are trying to see if they can identify the key organisms that you find in feces from a healthy donor and grow those up in the lab and basically put them in capsule form. And this, I think, is kind of cute, repopulating the gut. And there have been some hints of success. There have also been some failures in these clinical trials, but there's an awful lot of interest in that approach. So this is my last slide, and I apologize that I've gone over, but I think that now when we consider diseases, we have to think about us as the host, our microbiota, and various environmental factors, and we've talked about a number of these. We know that the microbiota is highly individual, varies over the course of a lifetime, but we can do certain things to really influence its trajectory. And I would say that diet is very important, and staying away from antibiotics as much as possible, equally important. But there's also, through development of brand new probiotics or fecal therapies, many other possibilities. And I think for me, this is incredibly complex, but what always seems to simplify it in my mind is when I go back and think about these studies in the context of an ecosystem. And I think we can go back and really remind ourselves what the field of ecology has learned over many, many decades. And so, just one other thing, this is, if you're interested now after listening to all of this, to know what's in your gut, there is this great project, the American Gut Project. Just Google American Gut. It's not terribly expensive. They will send you these swabs. You can sample yourself, you can sample your partner, you can sample your dog, and you mail the swabs back, and you get information that tells you how you relate to everybody else. This is me. I'm the big green dot here compared to everybody else who was in the American Gut study at the time. But you can see it tells you where you are, but we don't really know what that means. So I say, well, that's good. At least I'm part of the group. I'm not an outlier, but you know, I don't know what that means. But it makes a great gift. This was a couple of years. No, this was a couple of years ago. My husband asked me, "What can I get you for your birthday?" And I said, "Let me tell you. Here's your swab." And it was kind of freaky. We were both very, very similar. But you know, we eat the same thing. We probably share microbes. But this is the book that, and this is my last slide. If you want a book. There are lots of microbiome books out there, and I've looked at most of them. But this is The Good Gut by Justin and Erica Sonnenberg. This is solid science behind this. It explains some of what we've talked about. from a slightly different perspective, and they do give you some dietary recommendations. There are some books out there, you know, all these various microbiome diet books. I don't put a whole lot of stock in them because I don't think there's solid science behind them. But this I can highly recommend, and it's very, very readable. And this is a weird comic strip that I happen to like called Toothpaste for Dinner. And hopefully I have convinced you, at least in some way tonight, that your microbiota really should be considered as your new best friend. And with that, I'm happy to take questions. Thank you. Yes, first question right here in the second row. I saw that hand go up first.
Thank you for one of the best speeches I've heard here over the years. Thank you very much. My specific question has to do with Crohn's disease. Mm-hmm. Is there a specific bacteria that causes it? Are there specific bacteria that could cure it?
That's a great question. I've actually done a little bit of research on Crohn's disease. We see changes in some Crohn's patients where we see losses in these key anti-inflammatory bacteria. But there has been an idea circulating for a while, and there's an analogy with cattle, that Crohn's disease may be caused by a singular organism that acts more as a pathogen, a Mycobacterium species. And I'm getting ready to launch a study with some collaborators at Maryland to try and go looking for this particular bacteria. What I would say is it may be that both of those are correct, because Crohn's disease, inflammatory bowel disease itself, it's not a singular disease. As you know, Crohn's disease can have very different manifestations, and that may reflect different underlying etiologies. But not surprisingly, inflammatory bowel disease in its entirety has been looked at with a fair amount of scrutiny, but we're still without answers. And then I saw another question up here.
I'm probably not the only person here with artificial knees, but you know, when I left the hospital, I left with prescriptions for antibiotics to be taken anytime I was going to have any other kind of procedure by any other kind of doctor. It seemed to me that had more to do with issues of liability than it did about the possibility of any medical implications of these other procedures. I wonder if you'd comment on that. Yeah,
I want to be careful about commenting on that because that is outside my area of expertise. My husband has an artificial hip, so I bug him every time he goes to the dentist, have you taken your antibiotics? And he says, no, I don't want to take them because they don't, you know, I feel lousy afterwards. I think some of the wisdom is changing. I mean, I think to your point, I think there's very much a liability concern there. On the flip side, though, I will say that if you get an infection in a prosthetic joint and that infection moves into the bone, bone infections are terribly difficult to treat. So I think ultimately it sort of, in part, depends how much of a gambler are you. I mean, there's probably a low likelihood of that kind of infection happening, but nobody can predict. And but I am under the impression that this sort of wholesale thought that no matter what you have done, you need to take antibiotics is maybe being looked at. But I'm not giving you advice not to take antibiotics. Here. Let's wait for the mic.
You mentioned something about the birth canal, and what about adopted people who are, you know, at birth, who are adopted at birth, who the skin contact is now a different set of microbiomes? Oh.
Yeah, that's a really interesting question. I've never received that question before. Obviously, with adoption, there's going to have been an initial exposure to the microbiome of the birth mother. But over the first over the first 2 or 3 years of life, that microbiome is very unstable and almost seems to be trying on lots of different conformations until it figures out what it wants to be when it grows up. So while I'm not sure whose skin contact makes as much difference as whether it's a vaginal birth versus a C-section. And you think about it, once you say once you bring a baby home from the hospital and it gets passed around to all of the relatives and, friends that want to come and ooh and ah over a newborn, they're exposed as they probably should be to lots of different bacteria. So I'm not sure it really matters so much. Oh gosh, this is bad. I'm trying to give everybody in the room a chance.
Do levels of physical activity have anything to do with the biome, either positive or negative?
Good question, and there hasn't been as much done on that, but the answer seems to be yes. And so we can measure these differences. We don't know what they mean, but you might guess that physical activity releases endorphins and does all sorts of other good things, and that presumably would promote a more healthful composition of the microbiome, but nobody has looked at it in that level of detail. Oh God, this is awful. Somebody else choose the questioners. Stand up.
Vaccines and what they're made out of. And when you add our new diet that has genetically modified foods, how is this affecting our gut?
Well, those are two very different questions. I can tell you I've been involved in a couple of studies looking at live oral attenuated vaccines against intestinal pathogens. Now that's not, the measles, mumps, rubella vaccine, but these are vaccines, not unlike the polio vaccine that I got when I was a kid. I don't have children, so I can't speak. I don't even know what kids get for vaccines. We saw absolutely no effect on the microbiome in any of the individuals that we looked at. We've looked at 5 different vaccines. Now, these are not vaccines that are routinely given. These are vaccines in development to treat traveler's diarrhea or treat diarrheal disease in developing countries. So that's a segment of the vaccine world. Quite frankly, we were kind of surprised, and then we decided, maybe this is really a good thing that these vaccines, they're inducing the production of antibodies, but they don't seem to be impacting the microbiome. I can't say, and I don't know that I've seen any studies looking at any other vaccines. So I guess the jury is still out on that. And again, I'm not sure it would be, you would expect one result, but at least some of these preliminary studies suggest we're not seeing big differences.
And then GMOs?
GMOs. I don't necessarily think that GMOs are so terrible, but I don't know that anyone has looked at GMOs. Again, it's the number of papers that get published each year on the microbiome is enormous. It could be 10,000 papers a year. Nobody can keep up with all of that. I'm not sure. And again, GMOs are, when you talk about GMOs, there are a lot of things that fit under the GMO umbrella. So I'm not sure it makes sense to generalize across all GMOs. And I'm not trying to deflect your question. I just don't know the answer. Yes, back there.
When I went to the pharmacy several weeks ago to buy probiotics, there were probably 6 or 8 different type bottles on the shelf. How do you determine which is the better of those, or can you?
At this point, I would say you can't. We're really still in very much an empirical phase. Try ones, maybe they make you feel better, maybe they don't. And I think that's really the advice that a lot of physicians will give their patients. What I will say, what matters, if you believe that the presence of live organisms matter, which I think it does, you want to look at the packaging. You want to look to see if there's an expiration date. I think there is a great range of quality in the manufacture of probiotics. No guarantee that if you get a product that's well packaged is going to do something for you, but if you get a probiotic that doesn't seem to be, a lot of these come in foil blister packs and things like that, that's probably better than something else. But the science is still so early. And it's, you know, I happen to take one, I take a bifidobacterium only because I've been working on probiotics and I've read the scientific literature, and there seems to be good data in the literature to suggest that this bifidobacterium strain has some beneficial effects. But it may be doing nothing to me, but there is a placebo effect. I'm feeling pretty good every morning, so. Yes, over here.
If the fermented items, if they are heated through, the microbes die?
Yes, generally that's the case.
I read a book, I mean, I haven't read a book, but there is a book says eat dirt. Are we washing our foods, our vegetables too much, you think?
Well, that's, you know, that's an interesting question, because I would say if we weren't worried about pesticide residues, then yes, we probably are washing our fruits and vegetables too much. But I would certainly not recommend, nor do I, you know, take what I bring home from the supermarket and just eat it without washing. But you know, if I was growing tomatoes in my backyard and knew how they were grown, I doubt I'd run them under water before I slice them. And
do they cause gas?
Microbes.
Do they cause gas?
Oh yeah, gas is really important. Gas is a sign that you're alive. If you're not passing gas, that's not a good thing.
On that lovely note, let's thank our speaker.